viewers, welcome to my channel. Today I will discuss about glycolysis. I am starting from basic. Glycolysis is a metabolic process. So first of all we have to know what is metabolic process. Metabolism is a series of interconnected chemical reactions occurring within a cell, and the chemical compounds involved in it are termed as metabolites. Chemical compounds is called metabolites, and the whole process is called metabolism. Metabolic pathway is divided into catabolic pathway and anabolic pathway. Catabolic pathway is breakdown of larger complex, and anabolic pathway synthesis of compounds, that means formation of new components. Here glycolysis is catabolic pathway because here glycolysis means glyco means sugar, and lysis means breakdown. Lysis means break down. Then we can say that glycolysis means sugar breakdown. Then coming to the next slide, glucose. From glucose, how many pathways are involved? From glucose form two molecule of pyruvate through glycolysis by the help of 10 enzymes. Then from 2 pyruvate in anaerobic conditions form 2 lactate, which is fermentation to lactate in vigorously contracting muscle, erythrocytes, some other cells, and some microorganisms. In this case, lactate are formed. In anaerobic condition form 2 molecules of ethanol and 2 molecules of carbon dioxide, which is fermentation to alcohol in yeast. This ethanol is used to fermentation to alcohol in yeast. Then from acetyl CoA to form 4 molecules of carbon dioxide and 4 molecules of H2O, which going to citric acid cycle, and this cycle occur in animal and plant and many microbial cells under aerobic conditions. Then we will know what is glycolysis. Glycolysis is the first step of cellular respiration. It is an essential metabolic process where 10 enzymes catalyze reactions that covers glucose into pyruvate with simultaneous production of ATP. It is an oxidative process. 1 mole of glucose is partially oxidized to 2 moles of pyruvate. The major pathway of glucose metabolism occurs in cytosol of cell. This pathway occurs aerobically and anaerobically but does not involve molecular oxygen. Next, phases of glycolysis. Glycolysis leads to breakdown of 6 carbon glucose into 2 molecules of 3 carbon pyruvate. 2 phases are phase 1 preparatory phase and the phase 2 is payoff phase. Phase 1 preparatory phase 1 to 5th steps in which glucose phosphorylated by ATP to yield fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate then split into half 2 molecules of 3-carbon containing glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. Phase 2P of phase 6 to 10 steps in which release of 4 ATP molecules and 2 molecules of NADH during conversion of glyceraldehyde 3-phosphate to where there is a net gain of 2 ATP molecules per molecule of glucose in glycolysis. Glucose converted to glucose 6-phosphate. Here phosphorylation occur by ATP to form glucose 6-phosphate with the help of the enzyme hexokinase. Next, glucose and magnesium involved in this process. Next, glucose 6-phosphate converted to fructose 6-phosphate. This is a reversible process where magnesium involved and the enzyme involved in this process is phosphoglucoisomerase. Here isomerization occur. Then fructose 6-phosphate to fructose 1,6-bisphosphate form. Here also ATP is required. This is an irreversible reaction and fructose 6-phosphate is phosphorylated by ATP to fructose 1,6-bisphosphate. Step 4, fructose 1,6-bisphosphate then cleave to produce 2 molecules of 3-carbon containing, it is 3-carbon and it is also 3-carbon glyceraldehyde 3-phosphate which is also called GAP and dihydroxyacetone phosphate which is also called DAP. Here this reaction is catalyzed by the enzyme aldolase. Steps 5, isomerization. Dihydroxyacetone phosphate oxidized to form glyceraldehyde 3-phosphate. This reaction is catalyzed by the enzyme triose phosphate isomerase. Step 6, 2 molecules of glyceraldehyde 3-phosphate catalyzes the conversion of GAP, that is glyceraldehyde 3-phosphate, into 1,3-bisphosphoglycerate. Here NAD plus reduced to NADH plus H plus. Then step 7, 3-phosphoglycerate. Addition of phosphate to ADP to give ATP is termed as substrate level phosphorylation, as the phosphate donor is the substrate 1,3-bisphosphoglycerate. The product of this reaction is 2 molecules of phosphoglycerate. The remaining phosphate ester linkage in 3-phosphoglycerate is moved from carbon 3 to carbon 2 form 2-phosphoglycerate, and here the enzyme involved phosphoglyceromutase. Then dehydration of 2-phosphoglycerate. Here 2-phosphoglycerate is dehydrated by the action of enolase to phosphoenol pyruvate. This is a reversible reaction. Step 10. This is the last step, where is reversible transfer of high energy phosphoryl group from phosphoenol pyruvate to ADP. This reaction is catalyzed by pyruvate kinase. Here from pyruvate to 2 molecule of pyruvate is formed, which is 3-carbon compound. This is the whole cycle. You can see also this picture, which is a detailed pathway. 1, 2, 3, 4, 5, 6, 7, 8, 9 steps are shown here, along with the position of the carbon compound. Next, energy yield in glycolysis. Step number 1. You have shown that from transfer of glucose to glucose 6-phosphate, there is consumption of 1 ATP but no gain of ATP. Step number 3. From fructose 6-phosphate to fructose 1,6-bisphosphate, there is also gain of 1 molecule of ATP. Step number 7. From 1,3-diphosphoglycerate to 3-phosphoglycerate, there is gain of 2 molecule of ATP. And step number 4, from phosphoenol pyruvate to pyruvate, there is also gain of 2 molecules of ATP. Then total consumption of ATP is 2 molecule and gain of ATP 4 molecules. So net gain of ATP is 4 minus 2 equal to 2 molecule. Here you can see this, that here 1 ATP molecule, and step number 3 here 1 ATP, then 2 ATP molecule consumption, and step number 7 2 ATP molecule, and step number 10 here 2 ATP, then total 4 ATP, then net gain of ATP 4 minus 2 equal to 2. Okay, I am ending my class here. If this video is helpful to you, then please like, share and comment, and subscribe my channel. Click bell icon to get new notification. Thank you.